We recently asked a couple hundred of you emerging biotech leaders about your go-to sources of information when you face tough professional challenges. Your top response wasn't webinars, it wasn't scientific journals, it wasn't trade shows, it wasn't even consultants. Far and away, you said you most often turn to your peers for trusted insight. Enabling a community of peers is what the Business of Biotech podcast is all about. It's also what our new Business of Biotech newsletter is all about. Peer-driven content, no strings attached, delivered to your inbox once a month. Go to bioprocessonline.com/BOB to subscribe. The Business of Biotech is produced by Bioprocess Online, part of the Life Science Connect community, with support from Cytiva. Cytiva also demonstrates its commitment to the leaders of new and emerging biopharma at cytiva.com/emerging biotech. Check that out. Dr. Michael Hufford leads a biotech whose work sounds nothing short of science fiction. The company, LyGenesis, is working on organ regeneration in the incredibly high-need areas of liver and renal disease. And news coverage of the company seems to underscore just how next level these programs are. Headlines like, "Scientists look to grow mini livers for patients with organ damage," and "Liver grown in pigs' lymph nodes only serve to solidify the fantastical reputation of regenerative work." But scratch a layer deeper through conversation with the pragmatic Dr. Hufford, and the prospect of cell-based organ regeneration starts to look a lot more feasible. In fact, the company's got a phase 2A trial happening in end-stage liver disease right now. I'm Matt Pillar. This is the Business of Biotech, and on today's show, we're going to get to know Dr. Hufford and learn about LyGenesis and its allogeneic cell therapies that leverage the biology of the lymph node to engraft, proliferate, and form functional ectopic organs that recapitulate the native organ's function. That's a lot of words. Dr. Hufford, welcome to the show.
Thank you so much, Matt. It's a genuine pleasure to be here.
It's a genuine pleasure to host you. Those are some really fancy words I just used, and I'll tell you, I took them from your own material. Engraft, proliferate, and form functional ectopic organs that recapitulate the native organ's function. Anywhere you can use the word recapitulate, I think you're winning.
There you go.
We're going to talk about that effort in detail here in a little bit and the science behind it. But before we do, I want to talk a little bit about you and your interesting background pre-leadership of an emerging biotech. Ph.D. In clinical psychology. Did your clinical research fellowship in the Department of Psychology at Harvard Medical School. I don't know, 140-ish episodes in, I've maybe had a psychology, PhD in psychology or a psychologist on the show once, maybe. I don't know. I'd have to look back and dig. But I want to kind of catch up on like what was going on back then and how, what transpired from your PhD in clinical psychology, what transpired to lead you to the point where you're now leading a regenerative medicine company?
Yeah, sure.
That's a big question, right?
Well, and I'll try to keep it straightforward. I think, as is often the case, right, is this combination of both serendipity and then looking back on it, right, some of the skills that I acquired in my training to be a clinical psychologist. I kid you not, when you're in a meeting with the Food and Drug Administration, you got 20-some folks on their side. And as the sponsor, you're trying to carefully kind of navigate a course for a product forward. Having those sorts of listening and feedback skills to folks, in all seriousness, comes in, it turns out, remarkably handy. But
I can imagine.
But to back up, right? So I was very fortunate to train in an outstanding PhD program at the University of Pittsburgh. And it was very research oriented. And in particular, I was interested in substance use disorders, which also in the realm of psychology kind of firmly grounds you in the, biological underpinnings of our beings and the way in which, folks can use and misuse substances to kind of hijack that wiring. So you're always thinking a lot about biology. You're thinking a lot about the interface of people and biology and broader cultural, sociocultural issues around drug use. And so in many ways, I was just incredibly fortunate to have a wonderful education that way. But I also came through with training that was very unique to the pharmaceutical industry insofar as I had trained in capturing real-time data on symptoms, things like pain or pain relief. And it turns out that there's this, it's a niche literature, but it turned out to be very valuable to the pharmaceutical industry, that if you can capture data in real time, you can avoid some key drivers to things like the placebo response. And so I joined a startup that spun out of the University of Pittsburgh way back in 1999, before such things were fashionable. And our particular area of expertise was helping the pharmaceutical industry capture real-time data, or what at the time was called patient-reported outcomes, on endpoints that they were looking to validate as claims for indication. So this was a whole new world to me, right? As an academic, I was very frustrated with kind of the pace of academic research, and I found kind of the pace and focus that the intersection of business kind of brings to science, right? And so what I found myself is in a very fortunate position, having this kind of niche area of expertise, frankly, having a lot of both small pharma, but also large pharma with the need for this expertise and a need to engage the FDA in talking about novel, competitive, differentiating claims for products in development. So I found myself just a few years out of my professorship, sitting on scientific ad boards for small and mid-size and even a couple of large pharma companies, being asked to attend their FDA meetings, into phase 2 meetings, NDA meetings, to interface with the FDA, starting to present to the FDA about the science behind self-report and capturing data in real time. So kind of really accidentally found myself on a short path to getting engaged with the FDA in a way that had I taken a traditional role through pharma would have taken me probably a couple of decades, right? So so that, ended up accelerating my exposure both to drug development, to the world of regulation and FDA and the things they care about. So it really ended up just being a fascinating pathway. And as someone who enjoys science and enjoys telling the story of science, I had some great CEOs early in my career that also gave me the opportunity to kind of tell the scientific story behind companies and start fundraising. And so you had there the seeds of kind of how my career has unfolded, kind of that combination of, a niche scientific area of expertise, but understanding the FDA, understanding the need for fundraising, understanding a lot about clinical trial and clinical trial design to inform labels. So in that way, looking back on it, it was very unusual, but I wouldn't have changed,
What was did you have, I'm just real quick on this, I don't mean to belabor the point, but when you were studying, when you're in your PhD program and you were studying clinical psychology, did you have a career intention then? I mean, I'm thinking it wasn't necessarily what you're doing now, but did you want to go practice psychology?
So, I actually had a small private practice. So I was a practicing psychotherapist for a time. I actually got in a tenure track. I was fortunate to get a tenure track academic position right out of my internship from Harvard. So I was an assistant professor. I had my own lab. I had graduate students. I had started a small clinical practice. I was teaching both graduate and undergraduate work and doing my own research, but very quickly just kind of realizing some of the frustrations in terms of pace. Yeah. And what's always pained me, right? The unmet need that you see, especially when you work with patients that you're not able to help. And that stayed with me and will really for my entire life, not being able to help patients and being really face to face with the unmet need and appreciating that maybe academia is not always the best way to enable that change. And that I found, the whole biopharmaceutical industry to be, despite its public reputation, an incredibly noble and exciting enterprise to take new medicines forward. And so, I've been very pleased with my decision, painful though it was, 20 some years ago now.
Sure. Yeah, I can imagine. I mean, I guess I could draw more straight lines in my head between your academic work and pharma, like in the opioid arena, right? Sure. In pain management, and there's probably a ton of opportunity for guys with your background in that space. It's just I had to ask the questions because it seems like a bigger leap into true biologics and regenerative medicine. Yeah, sure. The other thing that kind of stands out to me about your background, you with the exception of probably spending some time in Boston, because I'm going to ask you about this too. You're also the interim CEO at Morphoceuticals.
Yes, that's
interim,
right.
Interim for some time. So
Yeah.
we'll get to that in just a second. But with the exception of probably doing some commuting to Boston for that role, you're a Pittsburgh guy. Like you're plugged in Pittsburgh. It appears as though you've kind of stuck a stake in the ground there. So I'm curious about that. Like what keeps you in Pittsburgh? Why are you rooted there? What keeps you there? And what does the biotech scene kind of look like to you?
Yeah. No, for sure. This is my third time living in Pittsburgh. So I have friends that joke I need to stop moving away. So yeah, I got my Ph.D. here, but then I spent some time in Boston on an internship, was a professor at the University of Montana, spent some beautiful time in Western Montana, as you might imagine.
Where in Western Montana were you?
In Missoula.
Okay. Yep. I've got a brother-in-law in Great Falls. We spend quite a bit of time out there. I love it.
Some great fly fishing out there and what have you. So then moved back to Pittsburgh for my first startup, but then, did a stint of, many years actually of drug development in San Diego at Amylin Pharmaceuticals and Cypress Bioscience. Moved to North Carolina, where I also got involved in startups and started my own company out there. And now I've moved back to Pittsburgh. So yeah, Pittsburgh kind of, setting aside the quality of life component, which I really do enjoy in Pittsburgh. It's an amazing multicultural place that's emerged from its Rust Belt days as really a totally transformed city. And folks that haven't visited are oftentimes genuinely shocked when they get here and they see the Google campus and the robotics startup scene out of Carnegie Mellon. And for me, both deeply appreciating the education that I got here. It's also the case the University of Pittsburgh gets, I think it's $700-800 million annually in NIH funding. It's an incredibly well-funded research engine there. And because of my background in the biotech scene, I'd been asked to act as an entrepreneur in residence at the University of Pittsburgh. And I mean, it really was just, an incredible orchard, right, that had come to fruition, where you have all this money pouring into the top of this funding funnel. And it's this place that's idea rich, but to a certain extent kind of resource and cash poor. So there's these amazing innovations that are being discovered and taken forward at the University of Pittsburgh. But Pittsburgh, because it's not Philly, right? It's not one of the main biotech hubs. It's certainly not Boston. It's certainly not San Francisco or San Diego or even Chapel Hill for that matter. It's the good and bad, right? The good is, as someone with, background forming companies and taking them forward, man, there's a wealth of opportunities here, right? On the flip side, it is not the case that you have a deep, executive management talent pool in Pittsburgh. Just a lot of folks haven't had that experience. And entrepreneurs that do start companies here ask me, who should I ask for money or where should I go? And I'm like, in Pittsburgh, you should go to the airport, right? It's like, you're not going to raise money in Pittsburgh. Yeah. You're going to need to get yourself to Boston. You're going to need to get out to San Francisco to JP Morgan. You're going to need to hit those areas. But like in our history as a species, has there ever been a time? I don't think there's ever been a time where it mattered less where you lived. Right. I mean, where you live, right? It doesn't matter anymore. It didn't matter that much pre-pandemic, and man, it sure doesn't matter now. So the extent to which lab space is cheaper here, you get a great quality of life, you have incredible intellectual capital, and then you can get on a plane to Boston, to San Francisco, to London. You can get the actual financial capital you need to move assets forward. And really where you call home, I would argue, is just increasingly not very relevant.
It's I was down in Pittsburgh not too long ago to visit Blue Sphere Bio. They're in a building that they share with some other biotech. New base is in the same building, right on the river.
Yeah, exactly. Yeah,
that stretch that they're in, right along the Monongahela. It used to be, to your point, super Rust Belt. Industrial. Yeah. There's quite a bit of development happening there, and from what I understand, quite a bit of that development, as I stated.
Oh, there's wet lab, there's small animal facilities. We have a lab in the adjacent building, actually, right there on the river. So no, the resources now, I think, the University of Pittsburgh and working with LifeX is working to incubate more life science companies. And you look at what Carnegie Mellon has done with the robotics scene here, right? This is the Silicon Valley for the robotics world. I mean, we had self-driving cars, years before other folks were talking about them right here. So, and yeah, I very much hope that the biotech scene will kind of continue to evolve and emerge here.
Yeah. Well, I share that sentiment to our listeners who have the opportunity to visit Pittsburgh. Please do. I live about an hour north, but and believe me, I'm in rural Western Pennsylvania. My wife likes to spend as much time in Pittsburgh as possible, shopping, food, and culture that we don't get up in the sticks. Yeah. So I mentioned Morphoceuticals. You still serve as interim CEO there. Is the work there related to what you're doing at LyGenesis at all, or?
I mean, yes and no. I mean, Morphoceuticals is a company, and in fact, this is Valentine's Day today, right? Tomorrow we will be announcing the close of an $8 million seed round at Morphoceuticals. And so,
congratulations.
First money in was through Juvenescence, which is also our lead investor at LyGenesis. And so, my investors had asked me to spend some time to help Morphoceuticals take its next maturational step. This is based on really genuinely pioneering work by Mike Levin and David Kaplan out of Tufts University, taking a bioelectric approach to limb regeneration, being able to regrow fully functioning innervated limbs in animals that don't normally regenerate limbs, but really even more fundamentally taking this approach that we're calling electroceuticals, the manipulation of native ion channel and gap junction modulators, really to trigger endogenous programs that we have within us, these bioelectric networks, present really from embryogenesis, right? I mean, the whole reason we have two symmetric limbs to start with is because we had a program where our cells could communicate to form limbs. And Mike and David's approach is really trying to hijack that biology and rerun those programs to exert remarkable therapeutic effects. And so Morphoceuticals, as our lead asset there, is in limb regeneration. And so certainly if you think of LyGenesis broadly as a tissue regeneration company, an organ regeneration company, at LyGenesis we use the lymph node and the natural biology of the lymph node as an endogenous bioreactor to regrow functioning tissues. And I'd say the thing they have in common is where Morphoceuticals isn't using the lymph node, it's using these electroceuticals to affect bioelectric patterns that are innate in our anatomical structures. They share, I think, a commonality, which is also kind of a theme in my own drug development experience, which is when you work with nature, you tend to have far more success, not just in terms of efficacy, but in terms of safety and ultimately with regulators when you work with nature rather than trying to trick it or work against it, right? And so if you think of our work at LyGenesis using the lymph node, we're doing the same thing that cancer does. Cancer probes its environment. If it finds the lymph node, it finds that a very hospitable home for tumor growth, right? Because the lymph node is this well-vascularized space that helps naturally nurture our T cells, but can be used for other tissue types. So Eric Lagasse, my chief science officer at LyGenesis, discovered that, we could put a number of different cell types within the lymph node and exert remarkable therapeutic effects, regrowing fully functioning organs that can actually exert life-saving effects. Likewise, Mike Levin's work at Tufts and Morphoceuticals is using endogenous pre-programmed connections and manipulating ion channels and gap junctions to re-trigger some of that bioelectric program. But again, working in a way that's very consistent with nature. And I think when you do that, we're showing that you can exert remarkable therapeutic effects and avoid some of the safety and adverse events that, as you know, right, are by far the most common killer of drug development programs is untoward safety events, right? So.
Yeah. Yeah. Well, I mean, and I made it sound like liver and kidney regeneration is sci-fi. We're talking limb generation at Morphoceuticals. We're going to do another episode on that.
You should have Mike and David on. That'd be a very enjoyable conversation.
I wrote their names down as we were speaking.
Yeah, Mike Levin, David Kaplan.
Yeah. So I want to talk. You mentioned just Actually, the first time you and I chatted, you talked a bit, you almost lamented. I don't want to use, lamented might be a strong word, but you talked a bit about sort of the pace of progress and the perception around regenerative medicine. And I want to kind of get into that. I want to get your take on that. You guys are working on these cell therapies for end-stage liver disease, end-stage renal disease, type 1 diabetes, to name a few. These are big, super important indications with very large, in some cases, patient populations. Cell-based regenerative medicine seems, a likely path of pursuit. And yet, as I mentioned, you sort of have lamented the pace of progress in regenerative, and acknowledged that it sort of had trouble cracking the mainstream. Yeah. So tell me about that. Like, why is that?
I mean, I think fundamentally, my own view is that, to a certain extent, you're caught in this dynamic tension between having to promise remarkable things when you're raising capital, because ultimately you need to entice investors to put capital at risk. And in biotech in particular, right, this is an incredibly high-risk business. The vast majority of things don't pan out, not just because biology is hard, but you're working in an incredibly, highly regulated environment. There's lots of challenges, right?
Yeah.
So lots of things don't work, which means to entice investors, right, you have to promise a lot and deliver even more. And I think sometimes as a field, what's happened is, especially, this isn't to blame academic founders, but I think especially when you have remarkable academic discoveries and people raising money based on those discoveries, if they haven't had the direct experience of taking things through the FDA and appreciating just how conservative, how painstaking that process is and how long it can take, it's just very easy to miss. You can overpromise and think, hey, we did this in my lab, I just raised some money. And you see this happen all the time, including by people that are doing deeply respected and well-deserved respect around their science, but they fail to appreciate really the chasm that can exist between the point of academic discovery and clinical translation. And so as a field, I think in regenerative medicine, there's been a lot of hype and there's been a lot of remarkable promise. You think of, how long have stem cells been discovered and around, or, the initial cloning of Dolly the sheep. And folks, I think, misunderstood then how early a lot of those discoveries were and how long the path was going to be to realize FDA-approved clinical therapies for some of those indications. So part of it is a tension that I think we all face, right, between generating enthusiasm for your work, attracting high-risk capital to it, right? And that when you miss or when you don't understand the time and cost, or you don't have a firm grounding in that, of the time and costs it takes to actually develop clinical therapies, you can end up as a field overpromising and underdelivering. And then, a few years on, folks like you and I are talking and lamenting the fact that, things are, oh gosh, it hasn't quite unfolded the way we'd hoped. And it is, I think there's an understandable and perhaps inevitable tension between those, but it's easy to overpromise and underdeliver when you're developing regulated therapies, for sure.
When you're striving to excel in a new arena, the best guides are the ones already doing it well. The Business of Biotech brings those voices forward to help new and emerging biopharmas turn their innovations, like mRNA and cell and gene therapies, into clinical realities. Tune in and subscribe for insights on hiring, regulatory, and other need-to-know topics for biopharma leaders. The podcast is brought to you in collaboration with Cytiva. Check out their resources at cytiva.com/emerging biotech. That's CYTIVA.com/emerging biotech. It's fascinating because I'm sitting here listening to you talk, sort of outline the source of that misperception as being not nefarious or intentional, which I see some bad apples now and then in this job. I observe, right? Like I observe people in roles like yours for a living and have discussions with them. And I've been caught up in conversations with some people who have ended up being bad apples who do exactly what you're talking about, create that hype, contribute to it to attract dollars in an intentional, nefarious way, because they know well and good what they're doing and that it's not quite baked, right? But you're talking about an academic phenomenon where, perhaps it's more naive than that. It's there's excitement around the science, and it's not necessarily with bad intent, but there's just some naive hype curve created that in the longer tail of things sets us back a little bit.
Yeah. Well, and to be fair, I think the blame is shared not just with academic founders that don't appreciate the path forward, but you look at some investors, right? Like folks, folks talk about, just in air quotes here, Silicon Valley investors, folks coming from tech and investing in the biotech space, right? Look, capital, it was Mary Lasker who said, capital is like frozen energy, and you unfreeze it when you pay people to work. So the extent to which folks are willing to fund biotech, man, I'll meet with anyone. It's the case though with some tech-based investors, because they are used to a pace of change and an ability to iterate and correct mistakes and release early demo products that have all sorts of, features or, bugs, right? But learn from that and still make money and still generate revenues and create incredibly successful businesses. And you hear this kind of trite saying like, oh, but biology's hard. It's like, yeah, like that's half of it. Half of it is, biology is really hard. It is orders of magnitude harder than a lot of what you're dealing with in a programming space. That being said, I also think I gave a talk one time and I showed a slide of the Wright brothers taking their initial flight, right? And imagining that it's easy to see biotech as like a technology problem. It just needs to take flight. But what's different is, imagine the Wright brothers were launching at Kitty Hawk, right? But they had to get full FAA clearance before they ever tried to fly. Like, imagine the bureaucratic paperwork of like getting permissions to try to do it. And then an FAA inspector comes out and looks at the plane and says, well, you're going to kill yourself. You're going to kill someone else. There's no way this thing's going to take off, right? Yeah. And I think what folks may not appreciate coming from tech, investing in the biotech space, is FDA regulations. And I want to take a balanced approach here too, right? They're incredibly onerous. It is incredibly difficult to take things forward. And if you haven't seen it and been in the trenches and been in those meetings with the FDA to understand how robust the preclinical sections of INDs, your investigational and new drug applications, need to be, how incredibly detailed your CMC, your chemistry, manufacturing, and control data needs to be, it's just easy to fail to understand that it's not just that biology is hard, but you're working in, I think, arguably one of the most heavily regulated industries on the planet. And look, we have the best innovation in the world here. We have the safest drug supply in the world here. So I'm not saying it's unnecessary or the FDA always gets it wrong. I don't think either of those things are the case. But the fact is, it is an incredibly onerous, highly regulated process. And whether it's Elon Musk at Neuralink or other folks that kind of jump into this field, not understanding how long it's going to take and how much capital that they're going to have to invest to see if it works. That, I think, is also a source of some of that tension between the promise versus the reality of developing these therapies.
It's a perfect segue. You touched on several themes that I want to ask you about, several topics I want to ask you about. So it's actually a very, very nice segue, because I want to get your perspective as the leader of this company, how that sort of environment impacts or affects your leadership of certain aspects of the business. And some of those aspects, I'll rattle a couple off that I'm interested in your perspective on, and you can start where you'd like to. Scientific development, for one, like, when you think about the metered approach that you're signing up for, due to regulatory constraints, due to investor, investor, I guess, appetite for getting behind a project and the realities of that project. You obviously have to meter your science behind that too. You're not going to go out there and tell investors like, hey, we need, $40 million. And by next February, we're going to have a fully functioning, regeneratively grown limb, right? Like, So science will be one. Like how does this environment impact your leadership of the scientific development? Another would be how it impacts your fundraising strategy. You know, who you're going after for how much and to do what realistically. And then of course the other would be regulatory strategy. So I know I'm bouncing around here a little bit, Dr. Hufford, but pick one of those and let's talk about it.
Well, and I think they intersect. And I mean, I'll tell you the kind of origin story of LyGenesis because I think it pulls at each one of those threads, right? So I meet Eric Lagasse. He spent the better part of a decade doing NIH-sponsored research with this amazing discovery of the use of a lymph node that can act as a bioreactor to regrow these functioning organs. Mm-hmm. He very smartly gets a patent on that. An issued U.S. patent. So that's a great starting foundation. He, from the scientific standpoint, had made the discovery, and very much to his credit, instead of just wondering how it worked, he wanted to see when it didn't work. He's like, how can I get this not to work? And when I first met Eric, he told me something I'll never forget. He said, he showed me these mice, this data in mice, where these gene knockout animals have a otherwise fatal liver disease. And he was able to rescue them in grafting hepatocytes, liver cells into their lymph nodes. The lymph nodes would eventually disappear, form these fully functioning ectopic organs that would rescue these animals. And it was just truly jaw-dropping stuff. He had just published in Nature Biotechnology. And it was much like a lot of professors I met at Pitt had done just groundbreaking work, absolutely incredible. And I asked what I thought was the question you wouldn't be able to answer, just jumping into the chase. I said, well, that's great. Truly, this is jaw-dropping stuff. But have you done it in large animals, right? Because the FDA is going to want 2 species, small animal, large animal. And I thought for sure he was going to say no. He's like, oh, yeah. He's like, we've done 2 studies. We've done one study at the Mayo Clinic, an independent replication. We rescued all those animals. Did another pig study here at the University of Pittsburgh using a different model, rescued all those animals. He said, Michael, I can't get this not to work. and as someone that does drug development, right, like you just almost never hear that, right? I mean, literally hairs on the back of my neck stood up because this is someone who is leveraging a lot of natural biology to exert a remarkable therapeutic effect, had not gotten lost in the mechanistic questions that you could spend lifetimes understanding. He was just trying to break the model and he couldn't break it. He had partnered with our chief medical officer now, Paulo Fontes, who was a transplant surgeon trained with Tom Starzl, deeply connected to the unmet need in liver disease, having cared for thousands and thousands of patients. And so you had right there this remarkable technological scientific promise. You had a decade's worth of work that had already been done. I helped pull together a briefing book with all the work they had done. And we went to the FDA and we said, look, here's our plan. We think we can take donated but discarded organs. We can isolate their cells. We've shown we can do that. We can exert these remarkable effects. Here are all the mice that we've studied. Here are the various large animal work that we've done. But you do need a native liver disease. If you or I with a healthy liver were to get engraftments of these cells in our lymph nodes, nothing would happen. They need in a milieu where they're triggered to start dividing and vascularizing and proliferating. And so we need to do this in patients. So our first in-human study, we think needs to be in patients with end-stage liver disease. So that was a sound scientific and regulatory rationale. But of course, wearing your business hat, I also know that if you have FDA meeting minutes, kind of the currency of the realm, right? That, whether you're Pfizer or you're LyGenesis, when you leave the FDA with meeting minutes, it says, here's what we agree to, here's what we disagree to, because they always disagree to something. And then here's our understanding of your next steps. Now you can go to investors and say, hey, I'm not guessing. I need X million and this much time, and I'm going to be able to get an IND and be into the clinic. And so at LyGenesis, we got our IND on less than $7 million of invested capital. The industry average, is ridiculous, right?
I wouldn't even venture a guess, but
it can be north of it can be hundreds of millions of dollars starting from scratch. So we had a very capitally efficient, relatively rapid path to clinic. Now we still had to do an IND enabling study. We had to do an additional trial. It was complex. It was, it was very challenging, but still in the grand scheme, right? Understanding that you could have that FDA meeting, get that buy-in, execute your IND enabling study, get your open IND and move forward and be able to go to investors and say, I'm not guessing, I'm not hoping. Here are the meeting minutes. You can see what they told us. Here are the bids to run the study. And like you had said, part of it is just kind of a ruthless pragmatism, right? It's understanding as sexy and cool as the science is, if you want it to be a human therapy, you have to cross that chasm, right, between academic discovery and checking all those boxes. And that just takes a kind of ruthless pragmatism of just a lot of blocking and tackling, to use a sports metaphor. Take things forward.
Yeah. Well, you did a fantastic job responding to my question, because you answered the question, but also tied in some of the science, and that's kind of where I wanted to go next with the conversation, and that's the science of putting these therapies together, the science of sort of behind the manufacturing. You touched on it, right? So you're taking discarded but donated donor organs, and you're doing something with them to develop a cellular therapeutic. So take us from there. Tell us about how where the organs come from, and then what the process looks like from to get receipt of the raw material to creation of a therapeutic.
It is remarkable, because as I tell the LyGenesis story, it's so easy, including for me years on now, to get wrapped up in the science of it and to show people pictures of lymph nodes that have turned into, histologically indistinguishable ectopic organs from their native counterparts. It's so easy to get lost in that. And I think probably historically to date, I've personally probably undersold the key question you're getting at, which is really fundamentally, is this a good business though?
Yeah.
Like, is there a business to be made here? And what I think is as equally as compelling as the science is, again, I'm not sure, it may be a case better lucky than good, but we have a lot of things lined up in our favor at LyGenesis. First, we're using donated but discarded organs. And it turns out in this country, when it comes to livers, at least 15% of livers are donated but discarded. And that can be for any number of reasons, right? It can be from someone who had a fatty liver, and so a transplant surgeon rather is looking at that organ and says, it's not ideal. I think I'd like to wait because organs are constantly becoming available, unfortunately. And so they wait for a better organ, so that organ may be discarded. There is, of course, over 200 Americans die every day of drug overdose in this country. Those are typically young to middle age, otherwise healthy individuals. And so you also have a lot of organs that tragically become available through drug overdose. Often you're not sure how long since the person was discovered from when they had overdosed. Some of those organs are absolutely used every day for transplant, but some are not ideal, so they're not used. So we're starting with a natural resource where you have these organ procurement organizations. No matter where you live in the US, you live somewhere where an organ procurement organization governs that geographic area. And they're trying to make sure that if you're an organ donor, and I believe everyone should be, of course, they're going to make sure that that organ has the greatest chance of being placed. But again, at least 15%, sometimes more, of the time that organ's going to be discarded. So we're aligned with the organ procurement organization because they don't want those organs to go to waste.
Yeah.
And they're going to be discarded. And so now they can bring them to us. And in fact, they do. They approach us now. We get organ offers saying, hey, would you be able to use this organ? Okay, so that's one.
And the reason for the discard is not reason enough for you to deem it not usable for you.
Now, it could be, right? There could be a physical damage to the organ from a motor vehicle accident where you're like, yeah, we just can't use it, right? But because what we're doing is not engrafting a lobe of the liver, what we're engrafting are really seeds that are going to grow your own ectopic organ.
And in most cases, those seeds are not affected by perhaps the drug overdose or the fatty liver disease or whatever.
Exactly. So if I'm engrafting you with a few hundreds of millions of these hepatocytes, but each liver is made up of billions of hepatocytes, the math's just in your favor. So traditionally, right, one donated organ treats one patient. Using LyGenesis' approach, one donated organ can treat 75 or more patients because we're taking each organ, we're both chemically and physically manipulating it to extract those hepatocytes, isolate and suspend them. That takes us about 6 hours Takes 3 to 4 people about 6 hours to go from a donated organ to having the cells isolated and ready to treat dozens of different patients, right? Now, the chemical reagents we use, it's a complex process. It's over 70 steps. It's part of an approved drug master file with the FDA. So while technically complex, relative to CRISPR and CAR-T and other, incredibly sexy, but frankly also staggeringly expensive technologies, this costs about $15,000 in reagents, and it takes 6 hours.
Yeah.
So you start with a donated organ, but discarded. You take 3 people, really in any GMP facility. So any CGMP facility, I tell folks we can bolt on to any existing facility. I mean, just to give you a sense, we need 10 feet of bench space in a hood, right? Like the technical infrastructure requirements here are really remarkably small relative to other like CRISPR CAR-T sorts of approaches, right?
And I don't want to get too far ahead of too far ahead of our skis here, but it occurs to me that with the prevalence of so many indicate liver, as we're talking about the liver right now, but also, kidney and down the road, other organ indications. That mobility, I think, would, if I'm not, like I said, jumping the gun, that's got to be key down the road too, right? Like the distributability.
Yes. It's like a word. Yeah. I thought, let's make it one. I think it's sort of a hub and spoke model. And in fact, what we've been doing at the facility we're currently isolating cells at for our clinical program, we've established that we can ship those cells within 4 hours of that facility, and we can ship the substrate, which just requires a few additional steps to process, 48 hours from the facility. So it's also the case that we can set up the LyGenesis program at a handful of these centers across the U.S., and you can service geographically the entire U.S. with just a handful of centers, which both assures quality control, assures just a good business model on the per patient license that you have. But then lastly, so each donated organ treats up to 75 patients, but we engraft our cells using an endoscopic ultrasound. Right. So we're going down through the mouth of the patient with this outpatient endoscopic procedure. The endoscope kind of transverses the gut, if you will, and can image along its the gut wall. When we identify a lymph node, a 25 gauge needle goes through the gut wall into the lymph node. We inject 1 milliliter of our cell suspension cell therapy, extract the needle, pull the endoscope back out. Whole thing takes about 10 minutes. And if you look at the cost for an organ transplant, so the acute in-hospital costs for a liver transplant today are just about $1 million.
Yeah.
CMS bills a outpatient endoscopic procedure at about $2,000, right? 10-minute procedure, light sedation, means a couple of things. Sicker patients that would not be eligible, which is what our first clinical trial is, for patients ineligible for organ transplant. Again, about 50% of patients who need a liver simply don't qualify. They will not survive the procedure, so they never even make the list, right? Yeah. So 50% of the patients never make the list to start with. Now, instead of having to undergo an incredibly large, major operative procedure, which is organ transplant, all they have to do is be healthy enough to undergo an outpatient endoscopic procedure under light sedation, right? So if you think about just on the strict business of it, right? You have this cell source that's readily available. You can isolate it and suspend your cells, making dozens upon dozens of treatments from each available organ. And then the engraftment procedure only costs a few $1,000s.
Yeah.
All that is the economic underpinnings for an incredibly successful and I think commercially viable therapy, because the pricing headroom you have, given the remarkable therapeutic effect you're having, like we talked about how expensive cell and gene therapies can be.
Yeah.
And when it's costing you 6 figures to develop them on a per patient basis, you're in for an incredibly expensive therapy. If it's only costing us a fraction of that for this cell therapy, it means you could have almost software-like margins for a cell therapy, which is, to my knowledge, pretty much unheard of, right?
Yeah, that is incredible. And to your knowledge, are there other, approaches to the same indications that you would consider competitive? And I don't want to talk about like you said, CRISPR and super expensive to develop cell therapies. Like I understand the difference here, but like sort of in the same, at the same or relative price point.
I'm not aware of, I have yet to see a company that has the same COGS, just the raw cost of goods, that LyGenesis has. I mean, it really is remarkable. So we're certainly aware of folks trying to use CRISPR, right, to do xenotransplants. You're trying to humanize pig organs. There you have all those costs, plus the full cost of a transplant procedure, right? So, I mean, just the economics of that are, I think, fundamentally going to be challenging. You had folks like Ambus that was trying to use like a rat as a bioreactor. Again, ran into a huge challenge with regulators and all of the FDA requirements around those xeno platforms, right? Taking one species to another. Yeah. That's incredibly challenging regulatory proposition. So we've taken the path of least resistance at LyGenesis by kind of doing the path forward that we just articulated. Look, it also means our first generation therapies are going to require immune suppression. That's not trivial. It's not trivial clinically. It's not trivial from a cost perspective. And so we are looking at different collaborations where we are joining forces with other folks that have some remarkable technologies to induce things like localized immune tolerance. Working with one company, iTolerance, has remarkable technology that they're using with pancreatic islets. We're collaborating with them with our hepatocytes to see if we can induce localized immune tolerance that would avoid the need for immune suppression. So that's some of the things in the LyGenesis pipeline that we're very excited about.
Yeah. Well, again, I characterized you as pragmatic from the outset of the conversation. The fact that you brought up, without being prompted, some of the challenges that you'll face is very much appreciated, Dr. Hufford. I've got a couple more questions for you. I know we're running short on time here, and I want to be respectful of yours, but I want to test, I guess I want to kind of pressure test your Wright brothers analogy that you referenced a little bit earlier.
All right. Okay.
I want to bring up the regulatory environment again.
All right.
It was a beautiful analogy. I think we'll find out as we pressure test a little bit, but it was a wonderful analogy. Is the state of what you're doing as unprecedented as the Wright brothers' flight might have been to the FFA that we can assume probably didn't even really exist at the time? So there was no need for the FAA. Which is, I guess, maybe the first point where the analogy might just show a little fissure, right? A little fissure, just a little crack. But assuming there was this regulatory body who, or the government kind of caught wind of the fact that the Wright brothers were among a handful of, skunk work operations trying to create an airplane that was going to fly through airspace. And they thought, we probably ought to keep an eye on what this little cottage industry is doing. Yeah. We can maybe make an analogy from the FDA to, some of the more progressive ATMPs or cell therapies, regenerative medicines like what you're working on. Is it as unprecedented as, you might have, as your analogy might allude to?
Well, so, right. The purpose of the analogy was right to highlight that tech investors are used to moving fast and breaking things, and that works because you can, you're breaking a computer program that has a bug.
Not taking a life into
Exactly. So for all sorts of good reasons, right? Look, I think one of the things, the benefits we had at LyGenesis in many respects is the pioneering work that Tom Starzl and others did in pioneering just full organ transplant, right? That was a huge, you want to talk about writing remarkable coattails. I mean, the story of the pioneering work that Starzl and others did around liver transplant, once you've gotten the FDA to acknowledge that, you can put a whole organ in someone and that can be okay, relatively putting a few cells in a lymph node, they had all sorts of specific questions around that. Yeah. But the precedent was set where it's like, okay, we kind of know how to think about that. I think morphoceuticals and like limb regeneration and using that electroceutical approach, again, for the FDA, it's about risk benefit. If you're treating someone with the amputation and you have the potential promise to regrow their limb, that's a remarkable benefit. And then you just have to look at the risks and the extent to which we're using, in that case, some repurposed drugs. We may also be developing and using it as a drug discovery tool, as a platform really to discover new drugs. But still, I think the risk benefit is going to be remarkably in our favor at morphoceuticals as well. So, part of that is just kind of being very thoughtful about regulatory precedent, being clear that the science is incredibly sexy and exciting, and that's what draws the capital. But to be good stewards of that capital, you have to have a very clear sense of how you're going to use that money to advance it through the FDA, which is a Byzantine, very complex, slow-moving bureaucracy. that also show up to work every day wanting to advance therapies, wanting to advance cures, but want to do it in a way that protects patient safety. And you just got to find a way to work with them in that context.
Yeah, very good. Okay, well, in the waning minutes that we have left here, why don't you give us an update on your clinical plans or progress?
Yeah, so at LyGenesis, we've now opened a second site in the greater Boston area. And so we are now recruiting patients with end-stage liver disease who are otherwise ineligible for transplant. We hope to be doing our first engraftment of our cell therapy to help them hopefully regenerate ectopic livers to help with their end-stage liver disease. So that is very much an open project with IRB approval, and we're actively enrolling those programs. So we will certainly be keeping everyone up to speed as we get those engraftments underway and that program moving forward.
Good deal. Yeah. Well, we'll be paying attention. It's fascinating stuff. You're a pleasure to talk to. I really enjoyed the conversation.
It was a real pleasure. It was a real pleasure.
Yeah. And next time I'm down the Pittsburgh area.
You please. Absolutely. Yeah, for sure.
Get together and have an Iron City beer or something.
Exactly right. Exactly right.
Thanks for joining me, Dr. Hufford. So that's LyGenesis co-founder and CEO, Dr. Michael Hufford. I'm Matt Pillar, and this is the Business of Biotech. We're produced by Bioprocess Online in partnership with Cytiva, whose commitment to new and emerging biotech is on display at its virtual biotech accelerator, which I encourage you to visit at cytiva.com/emerging biotech. I also invite you to visit bioprocessonline.com/BOB, where you can sign up for a monthly newsletter that's exclusive to Business of Biotech listeners. And while you're online, please leave us a review and some feedback. Share the podcast with your colleagues. And as always, thank you for listening.